Good morning, everyone. Welcome back to our seminar series on Translational Biomedical Engineering. Today, we have Dr. Hadj Henni, the CEO and co-founder of Rheolution Institute, who is going to talk about their novel technology on non-invasive measurement of mechanical properties of soft materials. I'll let Anis talk about the technology. But before we start, I have a few housekeeping notes. As usual, if you have any questions during the talk, please write your question in the Q&A box. You can find the box in the panel in the bottom of your screen. Make sure that you ask the questions in that box so we don't miss it if you write it in the chat box. Also, we always welcome your feedback. Please participate in the poll and provide your feedback to us to improve the quality of these seminar series. So also our next speaker is Dr. Malgorzata from the Netherlands. So her talk will be on 3D printing for regenerative medicine, from advanced materials to advanced designs. I'm very excited to host Malgorzata next week. Also, we have a list of fantastic speakers, upcoming speakers for this spring. So please feel free to visit us on follow us on our Twitter account, and then also email us if you have any questions regarding the future speakers and then regarding this program. Last but not least, I would like to thank our sponsor, Montreal TransMedTech, who has been with us since the very early stages of this program. So we would like to thank Montreal TransMedTech for supporting this program. So with that, I would like to invite Anis to give a talk on his career path starting from the graduate studies, a Ph.D. program, until becoming a CEO and starting a successful company. So Anis is graduated in civil engineering from the National Polytechnic School of Algiers, from Algeria, and holds a Ph.D. in mechanics from Université de Bordeaux in France. After a successful career in research, Anis co-founded Rheolution in 2009, and since then the company has grown rapidly and now is developing devices and technologies for characterization of mechanical properties of different materials in different sectors. So as CEO, Anis oversees Rheolution's development strategy over the Life Sciences and Agri-Food business units. So these are the main areas that the Rheolution, my best knowledge, is focusing at the moment. So I would like to thank Anis for participating in this event and then giving a talk. We are very excited to hear about your experience as an entrepreneur. You developed the technology during your PhD, and then you brought this into market. So we would like to hear your background and experience, and then also some of the new exciting developments that you have made. So with that, welcome, Anis, and then the virtual floor is yours.
Thank you so much, Mohsen. Hi, everyone. Thank you so much, Mohsen and Oman, to having me today. I'm very happy to participate and give a talk to show our experience and vision in our field in connection with translational biomedical engineering. So my name is Hadj Henni. I think Mohsen introduced me very well. I'm the co-founder and CEO of Rheolution. Today, I had a discussion to know where to go through this talk, actually. And of course, you're right, Mohsen. I will elaborate on the entrepreneurial journey of Rheolution, how we founded the company and where we are now today. But I also wanted to give an overall overview of where the science of material testing in the field of bioengineering applications are today. And my presentation will go through this different aspects. I will go through a historical view or overview where we are today and what are the challenges facing the biomedical community to overcome some challenges due to the limitation of traditional instruments. So I will share my screen with you and share my presentation. Do you see it?
Yes, Anis. Yes.
Okay. All right. So the talk today, my talk today is entitled "Translating Material Testing into Biomedical Applications Through Soft Matter Analytics." I will elaborate on the concept, the new concept of soft matter analytics, and as I said, I will go through a small historical overview of mechanical testing and engineering in connection with biomedical engineering to understand where the science and technology is today and what we are trying to do here at Rheolution to go to the next step in terms of material characterization. So let's set first the problem. To understand the problem, we will take a look back and see how material and mechanical engineering has followed the human evolution over the ages and centuries. So material and mechanical engineering started in a very primitive way to build typically Greek buildings, temples, monuments, and military buildings as well. So the engineers at that time, if we can call them engineers, and actually they are, they used to design massive, huge buildings with a very thick and stiff material like stones, of course. And the evolution of material science and mechanical engineering evolved with time, with the accumulation of knowledge, of science, of course. And today, the same science allowed us to build great bridges, public buildings, skyscrapers, and so on. So material and mechanical engineering first started in the, I would say, civil engineering world to build building and public buildings and so on. But with the evolution of science and knowledge, the material science and mechanics enabled also the emergence of modern, more sophisticated applications. I can mention over so many examples the technologies that allowed to explore space and atoms in the very small scale of the atoms and molecules. And also that science helped with other sciences and other engineering fields to develop new conversion devices and installation of energy, like wind conversion energy systems we can see here. The same science and the same technology also allowed recently, quite recently, since I would say 1 century, the development of medical devices that restore some essential functions in the human body. We can think about stents, knee restoration systems, and implants for heart and cardiac applications. These devices are now widely used, are usually stiff, inert, and in many cases passive. Actually, they are not dynamically interacting physiologically with the environment. And if we look at this evolution, we are now, a lot of thinkers, a lot of thinkers in history and strategy are describing the next revolution as the biorevolution, where actually science and research, both public and private, is investing a lot of resources, money, and talents to develop new functional, soft, and personalized tissues and medical devices to actually repair organs and tissues in the human body. This is a huge field of research, investigation, research and development. We do see the first applications. There are so many. I just mentioning here a few of them. We can mention, for example, bioengineered urethras grown from patients' own cells. This work has been done by the Wake Forest Institute in the US. We can mention bioengineered human heart, bioengineered vascular grafts, and also bioengineered skin, among other applications. So we can see here very easily all the potential of this field of research, investigation, and R&D in terms of impacting how it will impact the human health in the next decades and years. The promise is simply huge, and we can, let me come back to this slide, and we can see, and you all know how complex it is to develop bioengineered tissues. It involves a lot of science, of technology. We speak about biology, chemistry, physiology as well, material science, and also mechanical engineering, because mechanical and functional properties of tissues and organs are strongly linked, as you know. And interestingly, when I was preparing this presentation, I looked at some statistics from PubMed, and I looked at the occurrence of keyword bioengineering in the scientific articles on PubMed, and we can see here the evolution, the trend actually of this bioengineering keyword. It's increasing in a very significant way. It's typically exploding. And if we look at the trend over the last 40 years, we see that it's quite new in terms of field of research. The increase started in the early 80s, and we see here now truly a huge increase of in terms of interest and publications in this new field. In parallel, when we look at the keyword mechanics, we can see here the trend. The trend is going the same way, so it's increasing very significantly, but the increase started later. We can see that the increase started here 20 years ago, actually, and since 20 years, the interest in mechanics and in connection with medicine and bioengineering is increasing hugely. There is a delay between what we see here in terms of bioengineering and the interest we can notice here from the early years of this century regarding the mechanics in connection with bioengineering applications. So the reason for that is that the two fields are highly connected, and among all the engineering techniques and fields involved in terms of to develop new tissues and organs, mechanics is also one important aspect. We all know today that some diseases affect the stiffness of tissues and organs. Think about, for example, breast cancer, where actually the traditional way to detect is palpation, to detect the breast tumor. So we see here clearly that the pathology is affecting the mechanical properties of the tissue, and the mechanical properties can be used to detect pathologies. We all know that cell differentiation in tissue engineering is affected by the stiffness of the surrounding matrix, and it's very important to completely master that aspect of mechanical characteristics of the matrix in order to make things happen in terms of cell differentiation and growth. We also know that mechanical properties are good markers for the diagnosis of some pathologies. I mentioned breast cancer, but we can think about cirrhosis for liver and other pathologies. And we know also that bioengineered tissues and devices need to meet certain mechanical properties to be effective. A typical example is vascular tissue and vascularization. So if we design, for example, a bioengineered vessel, we need to make sure that the vessel is elastic enough, deformable, so we can adapt to the pressure of blood in the system. Otherwise, if we are developing a rigid vessel, we are seriously compromising the function of that tissue. So mechanics is, again here in this example, highly related to the effectiveness of bioengineered tissues. So now the question is, we know that mechanical properties are important. They are affecting the functions of bioengineered tissues. They are used to characterize and detect some pathologies. We all know that. So very rapidly, we are facing a question on how we can measure the mechanical or viscoelastic properties of biomaterials. When I say here during my presentation mechanical or viscoelastic, they are actually used interchangeably, and they mean the same thing. Viscoelasticity and mechanical properties in terms of in the context of soft materials are referring to the same thing. So the question is to know how we can measure the mechanical properties, because without measuring that properties, it's simply impossible to master them and to make the connection with physiological, biological aspects that are involved in the process of designing a tissue or bioengineering a tissue and producing it. So traditionally, testing technologies, we can sort them in two families. The first family, the oldest family, relates to compression testers. So the concept here is very straightforward. We apply a stress, a force on a sample, and we see how it deforms to get the mechanical properties of that sample. In the same way, we can apply traction to deform the other way the sample, and the same, we get the stress, strain, and from there we extract the mechanical properties of the material, typically the Young modulus and so on. This compressional testing techniques, or this family, has been used in the macro scale for objects in the scale of a few millimeters to centimeters, but it has been also used in the micro and nano scale, where we apply a force in the nano scale. Think about EFM, for example, AFM. We apply a force and we measure the feedback or the resistance the forces or the tip is facing to map the overall mechanical properties of the object in the nanoscale. And the second family of instrument relates to rheometers. So rheometers again apply a force, but in this case they are applying a shear deformation to the sample. And again, this kind of instrument are able to measure the strain, the stress, and to convert this information to get the viscoelastic or mechanical properties of the sample. What we need absolutely to keep in mind here is that compression testers have been initially developed and designed to test solids, stiff, strong solids, metals, concrete, name it, plastics, and so on. What we also need to keep in mind to understand the evolution and the challenges we are facing today, we also need to keep in mind that rheometers have been developed initially to test liquids, viscosity of liquids. There are great tools to characterize viscosity of polymers, very thick and viscous fluids, and so on. And their capacities have been extended over the 50 last years to characterize in a certain way the viscoelastic properties of soft materials. But you will notice, and you are noticing, we notice very quickly here that there is a kind of gap between compression testers designed for solids and rheometers mainly designed and initially designed to characterize liquids. In between these two extremes, we have a range of materials we call usually soft materials, soft matter, where we are facing a kind of gap. The existing technologies are not designed to characterize that kind of material. So traditional testing technologies were developed typically between the 18th and the 20th century to mainly test, as I said, solid materials and liquids. However, it's very difficult today to make the connection between, to use this kind of technologies in the context of bioengineering, where a lot of soft materials, soft tissues, and organs are involved. Not only, but a lot of applications are involving soft matters. So we said, and we need to keep in mind that these technologies have been developed in the context of solids and liquids. And because of that, they are not adapted to the very nature of soft materials. We also know that it's quite challenging to use traditional instruments to test non-destructively under sterile conditions and over long periods of time evolving biomaterials. When you are culturing cells in a gel, you are trying to see how the gel and the bioengineered tissue is changing over time, how the mechanical properties are evolving over time. And because of the nature of this testing instruments, in contact and applying a direct stress and compressing the material or stretching them, we're likely to destroy them when testing a gel or a bioengineered tissue. And it's quite challenging to keep the material sterile when we are able to keep it non-destroyed, actually. And finally, the use of this traditional technologies requires a high level of technicity. And I don't know if among the assistants, some of us have used traditional instruments, but it can be very challenging and difficult. Mainly, what we can very easily and quickly notice when we are using a testing instrument, compressional testing instrument or rheometer, they have been designed to be used by rheologists and mechanical engineers. When we are a chemist, a biologist, a material science, it's quite challenging to generate viable data using this kind of technologies. But the main limitation, I would say, of these traditional technologies relates to the fact that they make it difficult to correlate the mechanical properties of biomaterials with their biological, chemical, physical, and even physiological environment. That's a major point, a major limitation common to all testing technologies, and I will come back to this specific point because it's at the heart of our development strategy and what we are proposing to the biomedical community in terms of perspective in making the connection between the properties of their bioengineered tissues and biomaterials with their environment in terms of biological, chemical, physical, and physiological environment. I will come back to this point later. So this brings us to Rheolution's entrepreneurial journey. And so I co-founded the company with my partner, Cedric Schmidt, in 2009. And the story actually started in the context of research. So we met, Cedric and I, in 2006. And I started a postdoc actually in a lab here in Montreal, where Cedric was doing his PhD. And we worked together on a super exciting subject, developing shear wave elastography imaging. So shear wave elastography imaging is a medical imaging technique where the idea is to map the elasticity of soft organs and tissues in order to provide markers to detect and diagnose pathologies. So the idea is to use ultrasound. Some techniques in this field use MRI, but the idea typically is to use ultrasound to generate deeply into the tissue a kind of shear wave. Think about earthquake. So we are generating deeply in the tissue a shear wave that propagates, and we use the same ultrasound probe to measure how the shear wave is propagating inside into the tissue, and how that shear wave is interacting with potential masses or pathologies. So by doing so, by tracking the evolution of that wave, we're able to convert that information using physical and mathematical models into a map where you can see it here. For example, we can detect and localize a mass having a different stiffness compared to the surrounding medium here or tissue. And the technique is also used to measure actually remotely or assess the actual stiffness of the mass. So ultimately, the idea is to use it as a marker to differentiate between benign mass and tumors, for example, and so on. So we have today some applications in the field, some major ultrasound scanners and MRI devices are already including and integrating shear wave elastography as an imaging modality in their ultrasound scanners and so on. So we worked on that context, with Cedric for almost 3 or 4 years, and we faced during our development, the need to mechanically characterize the soft tissues and materials we used to work with, and we went naturally to use actually compression testing instrument and Rheometers, because they are the traditional instrument and the gold standards in the field. So by doing so, by trying to use this instrument, we experienced how it was difficult and we experienced almost all the limitations of traditional testing technologies. It was difficult. We were, to be honest, we were not able to generate the data we were looking for. Typically, the shear modulus of the materials we work with. We used the compression tester. By the way, we managed to destroy that tester because the parameters were so complicated to handle and understand, actually, that we destroyed the cell force of that tester, and same with the Rheometers. It was simply very complicated to get the data we were looking for. So we understood through our discussions by speaking with experts in the field and colleagues and so on, that we are really facing a problem: the absence of a simple, effective technology, simple to use and effective technology able to characterize the viscoelastic properties, mechanical properties of soft materials, which could be actually accessible for everyone without being an expert in mechanical engineering or a rheologist. We should be able to use in the 21st century a simple to use and effective testing equipment to characterize this kind of materials. And remember, we are at the beginning of a huge development process in bioengineering, and the needs in terms of characterization of soft materials, soft biomaterials, in our case, is going to increase over the coming years, because the research in the field is accelerating. So the need is really there, and we are facing collectively a problem. So we decided in 2009 to develop, Cedric and I, a new testing technology dedicated to soft matters. You can see here some vintage pictures of Cedric and I developing the very first prototype of our technology. So we started from scratch with a technology we have developed during Cedric's Ph.D. studies and my postdoc. We went through the process of applying for a patent with the Université de Bordeaux, discussing a license, and starting the business from scratch with a simple idea and a PCT application, a patent application at that time. So we spent a lot of time, as you can suspect, developing the technology, making that technology a product, a viable product, testing it with customers, improving the product, and changing it in the course of our development and interaction with customers and users, actually. So today, Rheolution is 10 years later, 11 years later. We spent, I would say, 6 years on pure R&D to develop the products. We now have more than 10 international patents, and we started the commercialization of our new products back in 2015. We had last year in 2020, despite the pandemic, plus 30% growth. And as I said, we're located in Montreal. We have an office in Europe, in France, where they are serving our European customers. And we have customers in North America, Europe, Asia, Oceania, South America, Africa, Middle East, and we're very proud of that. We are helping researchers, innovators from both academia and a lot from the industry, actually, better understanding the mechanical properties of their soft materials, and we are very proud of being a part of their success, because we are finally unlocking some critical information to develop effective medical devices and bioengineered tissues. So let's speak about the innovation a little bit. The innovation, our main product is called Elastosense Bio. You can see it here. And the Elastosense Bio is a benchtop instrument. It has the size of, I would say a desktop PC. This is the new generation of Elastosense Bio. We launched a few days ago, actually. It's quite new, and it's a non-invasive, modular, and easy-to-use instrument that measures non-destructively and without contact the viscoelasticity of soft biomaterials and medical devices for R&D, quality control, and process control in life sciences. We have included here a lot of innovations in terms of hardware. Let me start with the hardware part. I will show you why it is innovative and how it's answering the needs or some major needs from our field, bioengineering field, and I will switch then to the software part, where we are bringing also and coming with we believe a major innovation in terms of testing, analytical testing instruments. So let me show you here the principle. So the technology is based on the use of vibration. It uses a sample holder. What we call a sample holder is where the sample is contained. So the first step is to load the sample. Oh, sorry. All right, here we go. I'll try to do it this way. Perfect. So we load first the sample. So it's completely contained into the sample holder, and that sample holder has on the bottom part a flexible membrane. That flexible membrane has two functions actually: to maintain, to keep the material in place. Quite often the material is in liquid form, initially in liquid, and then becomes a gel, a soft gel. And the second function of the membrane is to confer to the whole system enough flexibility to vibrate when we apply vibration. So we use vibration to interrogate the sample, and when it vibrates, we see it here, some waves are propagating inside the sample. We use a very gentle displacement of a few micrometers, so it's not altering or destroying the sample. We use a sensor, a height sensor, to measure the actual height of the sample in real time. So if the material is swelling, we will capture, the instrument will capture that and display it to the user. And the technology uses a laser here from the bottom to measure how the membrane is vibrating. And actually, since the membrane is perfectly known, because we know the actual height of the sample, the membrane vibration, as well as the vibration of the sample, will strongly and entirely depend on the viscoelastic properties of the sample. So by measuring that, the instrument is able to run some models and algorithms to get the G prime and G double prime, the viscoelastic, shear viscoelastic properties of the material. And we repeat that process. Let's say we are studying coagulation or adulation kinetics. We repeat that same process every 10 or 15 seconds in order to get the time evolution of the sample. The new instrument is, we redefine the way we're designing the instrument and the technology, and we're using Internet of Things principles. Thanks to that technology, we have developed a modular system. The system operates, so you can see here, one module and a tablet. So the system is operated and controlled remotely with wireless communication through a tablet. we have developed an app in order to interact actually with the instrument, and each tablet is able to interact and manage up to 5 instruments. So we really like that modularity. It's possible for a user to start with only 1 unit and extend the system to up to 3, 4, or 5 units, depending on how their needs are evolving and how their workflow is increasing. So it's a very nice modularity, and we can run multiple tests simultaneously on the different units that actually form the system. As I said, the sample holder is removable. So again, the test is non-destructive. We can put the sample, test it, and remove it gently contained into the sample holder. We can store the sample holder and the sample into, let's say, an incubator, for example, and repeat measurement of the same sample over time. The system allows for real-time measurement of swelling and degradation, which is very unique in our field in bioengineering, because usually, as I said, it's quite challenging to get this kind of data with using traditional instruments. The equipment, the testing equipment, the Life Sciences Bio includes also UV light photostimulation. We can choose between 3 different wavelengths, and we can also combine between the wavelengths, and we can change the intensity of each wavelength to photostimulate the sample in a very flexible way. And of course, we have included advanced temperature control. Typically, we can go from 4 degrees C to 70 degrees Celsius, applying ramps and programmable ramps to study how, let's say, thermosensitive gels and hydrogels are reacting to temperature. But as I said earlier, beyond this hardware innovations, we really need a comprehensive solution that meets the needs of the 21st century in biomedical engineering. It's very exciting and even important to have a grid testing instrument able to offer non-destructive features and a very important, a very useful modularity and simplicity of use. But still, we need to come with a comprehensive solution to help our customers and users to make the connection between what they are measuring and what they are developing. So if we look at that, we have developed the powerful concept of soft matter analytics. I will develop the concept by starting by mentioning that today, what all testing instruments are delivering is viscoelasticity, mechanical characteristics. So the user has access to viscoelasticity when testing a gel, a hydrogel, bioengineered tissue, any other material. And if we think of it, if I tell you, hey, yesterday, you know what? I developed a gel with an elasticity of 4 kilopascal. I'm super excited, very happy about that, but you are not sure to understand why it's useful and exciting for me. So you will very likely ask me, why, Anis, is it important for you to have a gel with that specific stiffness? And immediately after that question, the answer will relate to the function of the gel. I will explain, for example, that having a gel with 4 kilopascal in G prime will help my cells to adhere and differentiate and grow. So if we look at the viscoelasticity carefully, we immediately understand that what makes viscoelasticity important is the connection, the interaction with its environment. So viscoelasticity is related to chemistry. We change the formulation of a gel, of a biomaterial, or of a polymer. If we play on chemistry, that will likely affect viscoelasticity. When we look at viscoelasticity, we make the connection with biology. As I mentioned, cells will interact in a certain way depending on the mechanical properties of their environment. Viscoelasticity will strongly depend on physics. So we are changing temperature, we are applying light, UV light, and photostimulation to induce some behaviors, then viscoelasticity is likely to be affected by that physical environment and parameters. And also viscoelasticity is related with physiology. Think about the idea of detecting masses and tumors by using viscoelasticity and mechanical properties as a marker. So we really need to make the connection between viscoelasticity and these 4 parameters. We can imagine others, of course, but let's focus on these 4 parameters. Additionally, these 4 environmental conditions are likely to be interconnected. So you can see very easily through your experience, and here especially, how complex it can be to correlate all these behaviors with viscoelasticity and with mechanical characteristics, actually. So mechanical characteristics are only one part or one piece of a bigger matrix of interactions and even sciences interacting between them. And this is a reality of bioengineers today developing tissues and devices. So our conclusion is that viscoelasticity by itself doesn't matter, actually, not at all. It only matters because it interacts with the biological, chemical, physical, and physiological environment of the material. That's why we decided to develop the concept of soft matter analytics, with the idea of first collecting on the same platform, on the same environment, software environment, all the relevant data describing our biomaterial or biosystem. So in this case, I hope you see the screen clearly. In this case, for example, we can decide to create customized fields describing our material. For instance, we may decide to enter the pH of the material prior to gelation. We can decide, for example, to enter the concentration of collagen. Let's say our material is based or made of collagen and hyaluronic acid. We can enter the concentration of these 2 polymers. And we can also decide, for instance, to enter temperature to see how temperature is affecting the formation of the gel and the long-term behavior of our gel. So we are providing an interface, an environment where the user can collect physical, chemical, biological, and physiological, potentially physiological parameters in the same environment to make the correlation ultimately between these parameters and the viscoelasticity, or again, the mechanical properties of the hydrogel. And this is quite unique. Our objective is to provide an integrated set of tools to collect data, to display the correlations and make the statistics, and highlight, ultimately, tools to highlight complex interactions and even sometimes hidden interactions and dependencies between viscoelasticity and the different parameters I mentioned. This is the way we are going here at Rheolution. We are, as I said, proposing the testing tool, but also offering the solution to get the intelligence we need to correlate and value ultimately viscoelasticity. But because again, viscoelasticity alone doesn't matter. It only matters when we connect that mechanical properties with other parameters. So let me conclude on applications if, yeah, I still have some time. Very quickly, Elastosense Bio is used in different fields to formulate hydrogels in tissue engineering, to develop and assess the efficacy of hemostatic agents, to characterize 3D bioprinted scaffolds, and also to study drug delivery and drug release. I will go through two different examples. The first one relates to the long-term and non-destructive testing of cells-laden hydrogels, work done by a certain Mohsen Akbari, Dr. Akbari from University of Victoria. They have used the Elastosense to actually formulate a hydrogel in the context of developing a functional in vitro infected wound model for drug screening. So they used the instrument to formulate a hydrogel made of gelatin and transglutaminase enzyme, to induce gelation. And they played with the two parameters, gelatin and enzyme concentration, to formulate the right hydrogel having the right elasticity. They selected that gel, and they used again the same instrument, Elastosense, to see how the gel is degrading when seeded with cells, how the different compositions or formulations are changing over time, over 14 days. So depending, as you can see here, depending on the formulation of the gel, when we look at the time evolution of the mechanical properties of the same set of samples, because the test is done non-destructively, we can see here that the 10% concentration degraded very rapidly. The 15% concentration maintained a little bit its integrity over 3 days, but started to degrade quite rapidly, compared to the 20% where actually the mechanical properties were super stable over 40 days. And when we look at cells proliferation over time, we see here that clearly cells are proliferating the right way with the highest stiffness gel, and this is typically what the Elastosense is used to in this kind of applications. I will not detail the rest of the work, and I invite you to read the article. It's super interesting. The team used the tissue that they developed to test some molecules and antibiotics. And the second application I would like to present relates to non-destructive and contactless technique to assess the viscoelasticity of blood clots in real time. This work has been done by a team from McGill University, led by Professor or Dr. Nezhat Chouhan. And in this case, the Elastosense has been used to test the clot formation, blood coagulation over time. And this is very exciting. I will mention later why, but here you can see how blood can be introduced into the sample holder of the instrument. The blue part here is the sample holder that contains the material. And again, the instrument has been used to characterize blood coagulation over time, clot formation, and there are different conditions or different concentration of calcium chloride. So they changed the concentration of calcium chloride to see how the coagulation kinetics is affected, and they have been able to compare actually measurement from Elastosense with those from a Rheometer and TEG. TEG is the standard standard instrument in this field, used mainly in cardiac surgery. And we found that the elastosense is very sensitive to the formulation of the clot, to the formation, and how the clot is changing over time, and how it's composed in terms of calcium chloride. It's correlated to the rheometer, while the tag is relatively less sensitive than the elastosense. So it's very exciting, and this opens for us new perspectives in terms of translating our technology to a biomedical field. So speaking about translation, and this is my my last slide. It's nice and interesting to notice that we translated initially a biomedical technology to make a new analytical technology. We are now in the process of offering and developing soft matter analytics as a powerful lab intelligence tool that gives scientists and innovators the full picture they need to investigate and innovate. And we are also starting the process of retranslating again or back our analytical technology to the biomedical field through the testing of human blood coagulation. And I'm super excited to announce that we have been selected recently, 2 weeks ago, to achieve a project with McGill University in order to test the coagulation, using the elastosense test the coagulation of COVID-19 patients to see how it has been reported that coagulation issues can be a source of complications and sometimes, unfortunately, death. So the project will serve to characterize coagulation of blood coagulation from COVID-19 patients in order to see what kind of markers we can find and how we can anticipate actually potential complications in terms of blood coagulation. Thank you so much for your attention, and if you have any question, I will be happy to answer.
Thank you so much, Anis, for your great talk, and it was really educational also for young scientists, your journey and all these challenges that you had along the way, and the inspiring technology. So we have lots of questions in the question box regarding the technology and the science behind it. So Gosia, she's our next speaker. So next week, she's asking that how do you measure swelling and degradation based on which material property, for example, by change of stiffness? And the second question she has is that, is it possible to measure network relaxation time?
Yeah, I will, maybe I will share again my screen because that will help. Yeah, let me not, here we go. All right. So let me come back to this application. So regarding the first question, how can we measure the degradation of a gel? It's simply through following the evolution of its stiffness over time. So in this example, typically, what the team did was to follow over time the mechanical evolution over 14 days, the mechanical evolution of 3 sets of formulations with cells. And the first set here, you can see it is very stable, and it was not degraded naturally. The second set was degrading, so the shear storage modulus, which is the stiffness of the gel, was degrading and decreasing over time, which is a clear sign of degradation, while the softest material degraded very rapidly. And the way we monitor degradation is really by following over time how stiffness or we call it stiffness, but the right wording is shear storage modulus, is changing and decreasing over time. The instrument also includes a height sensor, so we are able to measure the height of the sample. So if the degradation mechanism is superficial, since we measure at each measurement time the actual height of the sample, if the sample is decreasing in volume, we'll catch that through that measurement, and you can display over time how the height is changing and decreasing while the degradation happens. In the contrary, if the sample is swelling or retaining the volume, the instrument will also capture the change in height and display in real time how the height is changing. Regarding the second question, it's a very good question. The ultrasound is not able, and we didn't design the instrument to characterize the relaxation of a material. That's a very important question. It is, I believe, very, very well addressed by traditional instruments. Rheometers or even compression or traction instruments are able to characterize that relaxation behavior, which is related to the viscoelasticity and the viscous behavior of the soft matter. We don't do that. We use vibrations, and when we induce the vibration of the sample, the way the sample or the vibration is damped over time is directly related to the viscosity of the material. So we don't see or look at the viscosity from the relaxation perspective. We look at that from the damping perspective. So the instrument delivers G prime, which is the shear storage modulus. It also delivers the shear loss modulus, double prime, which is related to viscosity.
Great, thank you so much, Anis. So the technology that you have developed and the new technology, Elasticity Bio, also triggered some discussion in the Q&A box. The scientists and the audience in the panel in the event is asking how much volume is required to run a test, and how much is the maximum. And they're debating, the volume here in the Q&A box. So can you answer how much volume, and if it's thick, the Elasticity Bio can measure the viscoelastic property? This question is from Migan from McGill.
Okay. Sure. Yeah. Sure. No, it's a very frequent question, and it's important because, especially in our field, materials are expensive, and we need to consider that aspect as well. So the sample holder can contain between 500 microliters up to 7 ml, and the reason, so it's quite large in terms of range, and the height we should use depends on the elasticity of the gel. So basically, when the gel is super soft, we need to increase volume in order to get the sensitivity we need to extract and measure the viscoelasticity of the gel. In the contrary, where the gel is stiff, very stiff, we can reduce hugely and significantly the volume. So the volume depends on the elasticity of the material we are trying to characterize. The other aspect we need to consider and keep in mind is the non-destructive, the fact that the instrument is characterizing the material non-destructively. So contrary to classical technologies, I would say, we're not losing the sample after testing. We're able to continue measuring the same sample over time. So even if we use on the Elasticity a little bit more sample than usually we are able to follow the same sample over time rather than destroying at each measurement time the sample and testing a new one actually. So that's also an important aspect, I believe, in from that perspective.
Thank you so much for the clarification. Duna is asking, oftentimes tissues as soft material need to be measured. Inherently, the tissue will have different properties when it is dead or live, detached or avascular, without the blood vessel, versus when it's part of an organ. What are your ideas or thoughts about these differences when you use your machine to measure the viscoelastic properties?
No, sorry, Uman, I'm not sure to understand the question. How often?
No. He's talking about different tissue properties when the tissues are live, or they have vasculatures, or the cells are dead in the tissue, or he's talking, that if the machine is able to measure the mechanical properties of the tissue, soft tissues, and in different, hemostatic stage of live cells or dead cells. He's wondering about this.
Yeah, I will recommend. Yeah, I mean, the instrument is delivering, and this with this question we come back to the soft matter analytics concept. The instrument at a very specific time point, when we are using it to measure a material, is delivering a result. The result by itself is not telling us a lot of things. We need to correlate that result and see how it's changing over time. So let's say you have cells in your bioengineered tissue, growing cells, and in the course of that process, let's say they are producing ECM. If ECM is produced, that will affect the mechanical properties of the construct. In the same time, if the cells are degrading the construct, we can also expect an impact on the mechanical properties of the overall construct. So when we use the instrument to follow over time how it's changing, we need to correlate the measurement with other techniques, let's say imaging or other assays, to know what is going in the microscopic scale, how cells are growing, are they still alive or not? If I notice stabilization of the mechanical properties over days, and we are not expecting that stabilization, it could be a sign that cells are simply dead or degrading. So I would say it's important to have a look on how the mechanical properties are changing over time while a biological process, for example, is happening, and to correlate that information with other data coming from other analytical testing, analytical techniques, or imaging. And it's by connecting these two sets of data through the soft matter analytics platform we have developed. It's by connecting these two sets of data that the meaning of what's going on and the explanation of the processes will appear clearly. I don't know if I'm correctly answering the question or not.
I think yes, and this was also the question from other audience, Likon. She was, or he or she had the same question, and also she's asking about the range of G prime and G double prime that this machine can measure. What are the ranges of these two modules?
Yeah, the range is going from, for G prime, goes from a few pascals, so typically 10 Pascal, up to 1 megapascal.
That's great. And of course, he or she is asking that did you or have you ever compared your results with the traditional machine and or cone and plate rheometer or others? So of course, yes, and then you can also elaborate more. So,
yeah, sure. We did that. We did that many times actually with partners and customers and so on. We have at least one or two published articles that show how the elastocence is correlated to Rheometers on non-biological systems. So yes, the instrument has been validated and compared with Rheometers, and that's super important. But let me, if I may be clear, we are not saying here that we don't need Rheometers and we don't need compressional testing. No, I'm simply saying that if we want to equip correctly and get a fully equipped lab, we will ideally have three technologies. A Rheometer for liquids and fluids, viscous liquids, an elastocence for soft matters, and typically for biological applications, and a compressional testing instrument to characterize stiffer materials. So we don't see our technology as directly competing with traditional instrument, but it's we should see it as complementary tool actually to characterize different kind of materials under different conditions, and to make the relation between the mechanical properties and the biological chemical conditions as well.
Thank you so much. So another question is about sample preparation. So Glikon again is asking, because we're working with the soft materials or liquids, so there's a chance of forming bubbles. Is there any, if there is a bubble in the samples or hydrogels, triggering any error in the measurement?
Yeah, this is a very precise question, and yes, it's a very relevant question, because if we untrap a bubble in the sample, it depends on the size of the bubble compared to the whole size of the gel. If the bubble is taking, let's say, 30% of the sample, that's definitely an issue. If the bubble is super small compared to the whole sample, that's definitely not a problem. So if bubbles are an issue, what we can do is to vacuum actually the sample into the sample holder. Remember, we can manipulate the sample holder without affecting or destroying the sample, and we can put everything in a vacuum to extract air. Yeah, that's the way. And of course, the way we are pipetting and introducing the sample into the sample holder is also important to avoid or minimize the introduction of bubbles inside the sample.
Great, thank you. So another question that is also my question too. Daisy is asking. So we are talking about cells, the cocktail of cells and bioinks or hydrogels, and measuring the mechanical properties. But he's asking a question: if we can measure the mechanical property of the cells only, we culture the cells on your sample holder and then measure the mechanical properties.
I think we are not. No, we are not there. We designed the instrument to characterize scaffolds, 3D matrices with cells or without cells as well, to characterize hydrogels. But when we look at the size of the cells and the constraint, I mean, I would recommend to use an AFM, for example, to characterize the viscoelastic properties or mechanical properties of cells individually, because the scale is so small that it doesn't make sense to to use a device designed to characterize the overall macroscopic viscoelasticity rather than the microscopic viscoelasticity.
Thank you so much. Again, he has a follow-up question about correlating or comparing this result with the tissue mechanic, formula or equation like Hall-Zapfel. It's I don't know if I'm pronouncing. I mean, because you already answered that you can compare with other traditional and conventional methods, but he's now talking about simulation or calculated soft tissue mechanics. If you already compare with those models in the literature, yeah.
No. The answer is quite straightforward.
Yeah. No. Okay. So Chris from University of Victoria is thanking you about your great talk, and he's also kind of applauding you because of this technology. You have lots of collaboration with the university and industry, and specifically Mohsen is sitting here. So can you please comment on the application of your technology in testing non-Newtonian fluids?
It's a very good question, and we answer it quite oftenly. The elastosense is not able, actually, it's very clear. It's not able to measure the properties of fluids, even Newtonian or non-Newtonian fluids. And again, the limit between non-Newtonian fluid and soft matter is very sometimes not clear, but we define the range of operation of the elastosense as the time where the material behaves mostly like a solid, a soft solid, but still a solid, rather than a fluid. And that happens when tan delta or G prime is higher than G double prime. So the elastic behavior of the material is, I would say, stronger than G double prime. Then the elastosense is able to characterize the gel or the forming material. And before that, we are clearly in the fluid liquid space where we can find Newtonian and non-Newtonian fluids. So the instrument is operating only when G prime is higher than G double prime, when the material is exhibiting a more elastic character or behavior compared to viscosity.
Great, thank you so much. So I think there is no other question in the Q&A, but Mohsen is a great example of user of this machine. So if I think he has many questions. So we can go ahead, Mohsen.
So you still have questions 3 years after, Mohsen?
I do. Well, the questions. Well, first of all, thank you very much, Anis. I really enjoyed your talk, and then I have questions. You always have questions about not the technology itself, but I have questions about the translational aspect of this technology. So when I'm thinking about these technologies that you're developing, and then how these can be used in clinics and eventually for in real tissues, there are a few questions that I wanted to ask. So the first question that I have is not is along the direction of measuring the mechanical properties of cells, but mostly not single layers of cells, but maybe just tissues. Have you thoughts? What are your thoughts about using the technology Elastosense Bio for measuring the mechanical properties of biopsy tissues, maybe skin tissues, heart tissues, and maybe some other tissues? So I wanted to hear your thoughts on those applications.
Clearly, the I do see the interest to characterize biopsy tissues. If I come back to the very early years of Rheolution in 2009, with Cedric, we have clearly considered that possibility to characterize biopsy tissues, and our I would say strategic thinking. The conclusion of the thinking was that it was not the way we wanted to go 10 years ago, because we are coming from a research field where people are trying to develop non-invasive techniques to characterize the viscoelasticity or mechanical properties of tissues. Biopsy is still, of course, used for other purposes, but characterizing the mechanical properties is, I believe, going in the direction, non-invasive direction. That's the first point. The second point, we made the decision 10 years ago to avoid going in the diagnosis field, and we made that decision because of multiple reasons, but the main one is that the entry barriers in the clinical applications is relatively high, and considering our objectives 10 years ago and the aim of the project, we decided to avoid that clinical space. 10 years later, sorry, we are definitely in a different situation, and we are thinking about coming back to the clinical world with a device that clearly answers clinical needs, and we are thinking more and more about blood coagulation as a potential application for technology to translate back the technology to the biomedical field. So that's where we are in terms of developing our technology and applications. Of course, if we go that direction, and we want to go that direction step by step to validate each step to secure actually our investment and so on. If we go that direction, that will take, of course, some technological improvement or to adapt actually the equipment or technology to make it a medical device.
Okay, that's great. One thing that I wanted to so that's for diagnostic application, and I understand your strategic decision why you didn't want to go toward that direction. But as you know, and maybe the audience also know, that is part of the field in biomedical engineering. They use patient-derived ex vivo tissues. So they use patient-derived samples for drug screening applications. So instead of growing the cells and engineering the tissues using cells and biomaterials, they just collect the tissue and then they just use that actual biopsy tissue from the patient in well plates, in dishes. So I was wondering if this technology, or if you see any application in that direction using this technology, maybe just collecting the sample tissue from the patient, putting it in the sample cups, and then maybe just applying a drug, just giving it a drug, and then see how the drug affects the stiffness of the tissue. Do you see any application?
Oh, clearly, yeah, clearly. I can tell you, Mohsen, the technology is able to characterize soft tissues ex vivo. The only constraint is to confine the sample to the shape of the of the sample holder. We have some constraints in terms of volume, depending again on the stiffness of the gel, but of the tissue. Sorry, but it's doable. It works. Now the challenge I can see, and that's very, very interesting in terms of research, how to keep the tissue alive when it's interacting with, let's say, drugs, for example. We didn't invest so far actually to develop a bioreactor, for example, or something like that to keep the tissue alive. But it could be a great collaboration avenue to develop such a bioreactor.
Good, awesome. So, well, thank you. So we have only 4 minutes, but I really wanted to ask this question. So I'll be quick. When we go, so I'm thinking about a material from realization. So you develop a material, then you want to test it in vitro and then in vivo, and then bring it into clinic to get the approvals, right? So from Health Canada or the FDA. So at some some stage, you need to perform some characterization studies following ISO standards, right? By the FDA and then Health Canada and other regulatory bodies. Do you think that Elastosense Bio can be used to characterize the mechanical properties of materials so that they can be accepted by these?
It's actually the case today. We have we have a lot of customers from the medical device space in the US and Europe. They develop medical devices. Some of them in the space of hemostatic agents, and they are already using the Elastosense to develop their material, to get their material certified by the FDA, and we're moving more and more to quality control applications, control the quality of their devices. So yes, it's already the case. The instrument is working very well in that context, and especially with the new version, we are meeting some important standards in terms of software compliance to FDA standard on securing, data and access to data and modifying processes and so on. And I think the new tool is even more adapted to perform R&D, QC, and quality assurance in a context of medical devices. It's already the case with a couple of customers, mainly I would say in the US. Yeah.
Okay. Great. I can talk about this more and more, but maybe later because you're running out of time. But thank you very much for your very nice presentation.
Thank you, Masen and Oman, for having me, and thank you for the assistance.
Thank you so much, and look forward for the next session next week, Wednesday at noon Eastern Time of Canada, with Professor Bigon from Groningen University. So follow up on us on Twitter and LinkedIn to get the information. Thank you so much, everybody, and have a great day or night. Take care. Bye-bye.
Thank you. Bye-bye.
Bye.